Hi, it's Dan Carlson, and welcome to another episode of INmune Bio. Today, we're with Parris Pope, who's the head of medical affairs here at INmune Bio. Thanks for joining us, Parris.
Good morning, Dan. Thanks for having me.
Appreciate you coming on here today. And maybe since you're new to the Inside INmune Bio videos, you can provide a little bit of your background and history if you don't mind.
Sure. So I'm a pharmacist, a doctor of pharmacy by trade. I've been in the pharmaceutical industry for over 15 years now, almost exclusively in neuropsychiatric or supportive neuropsychiatric drugs and research. Um, I've been with INmune Bio for a little bit more than 3 years. I joined the company in 2021. And I'm very excited to talk about what I consider one of the most elegant mechanisms in neurodegenerative disease right now, and that is the very selective neutralization of inflammatory soluble TNF within the central nervous system.
Great. We're looking forward to that. So, kind of the genesis of this interview was a paper that came out last week. Maybe you can talk a little bit, provide a high-level overview of what was the results that were released in that paper. They're pretty important.
Yeah, so that's you're right. That is a very important finding from these experiments. So to give you a quick background, what these investigators have done is they've utilized well-known and efficient models of inducing inflammatory demyelination. So that's the degeneration of the myelin sheath and myelinated tracts in the brain. And and these are old models, right? So this is the experimental autoimmune encephalitis model, which has been used extensively in the development of drugs for multiple sclerosis. And also the more acute cuprazone model, where you provide the cuprazone, which is a copper chelating agent, to experimental animals, and they almost immediately develop inflammatory demyelination. So these two models are well established, and they give you a very good platform for measuring the effect of a whatever intervention. In this case, it would be XPro 1595 for measuring the effect of an effective intervention on that inflammatory process and the outcome thereof. Um, so to give you a quick breakdown, once again, these are well-established models. Have been used for upwards of 30 years, and we have examples of XPro being effective in these models in previous studies, right? So the question, one of the key questions is, what's the difference in this model compared to the other models, right? So this process of demyelination, we talk about myelin, we're talking about myelinated axons. And this is white matter in the brain, right? It's called white matter because when you do an autopsy, these heavily myelinated tracks, appear white to the in a color spectrum. So we're talking about myelinated axons in or white matter in the brain. But also we have to understand that myelin is also critical in cortical gray matter as well. It doesn't show up necessarily to the naked eye in cortical gray matter, which appears gray. That's why it's called gray matter. But in cortical gray matter, you also have a certain level of myelination, and the same process, the same inflammatory process that is associated with demyelination in the white matter tracts also occurs in cortical gray matter. So these models give you a very, once again, very efficient way of analyzing the effect of not only the inflammatory processes, but a potentially effective intervention, in this case, XPro 1595, right? So the key takeaway here is that, once again, these investigators have induced a acute inflammatory process that results in demyelination of not only the white matter, but also in the gray matter. They've measured it here specifically in gray matter. And they've shown that they can impede this process by giving XPro 1595 through peripheral injection. And we'll talk about that in a minute. But they've given XPro 1595 peripherally and shown that they can inhibit not only the inflammatory process, but the outcome, in this case, demyelination. So again, just a very clean, in my opinion, example of what I consider an elegant mechanism of action, and that is the selective neutralization of inflammatory soluble TNF with XPro 1595 in this model.
The paper talked a lot about repolarizing of microglia. Can you talk about the importance of that, and what does that mean in layman's terms?
Yeah, so let me back up a little bit here and talk about activated microglia. Activate microglial activation is actually a natural process, right? So that activation of microglia are the primary innate immune effectors in the brain, in the central nervous system, right? So activation of microglia is known to occur in all neurodegenerative diseases. And in this case, we're talking specifically about Alzheimer's disease and multiple sclerosis. The problem with activated microglia is that the processes tend to get oversimplified very often, and to the point where we have begun to associate microglial activation with a detrimental neuroinflammation, which is not actually true, right? And so in the models in humans, and the models that are available now, and this is much more advanced than it was 10 years ago, what we see is a phasic increase in activated microglia as neurodegeneration progresses, right? So specifically to Alzheimer's disease, early in the process, when we start to see confirmation of aggregation of the abnormal protein that's associated with that disease. So now we're talking about amyloid beta. As the amyloid beta begins to aggregate, we see on PET scans and through fluid biomarkers clear signals for activation of microglia. And that first phase of activated microglia is generally associated with a beneficial process, right? So microglia are phagocytes, and that early activation is associated with beneficial increase in the phagocytotic process of microglia for the purpose of removing cellular debris, right? So the initial phase is an enhancement of the ability of microglia to more efficiently remove abnormal proteins and cellular debris. However, as these neurodegenerative diseases progress, we see a second phase of activated microglia, and that level of activated microglia is associated with dysregulated neuroinflammation and what eventually becomes detrimental neuroinflammation that is potentiating the neurodegenerative process that leads to end-stage disease, right? And so in this paradigm, your objective is to inhibit the detrimental neuroinflammatory processes associated with activated microglia without inhibiting or impeding the beneficial processes that are required for maintenance and repair of neuronal tissue.
Gotcha. So if I could put this in a nutshell, there's kind of, and tell me if I'm right or wrong here, there's kind of good inflammation, bad inflammation. Activated microglia can be good, they can be bad, and what we're doing is
right. Yes, exactly. Right. And so what happens with activated microglia, right? So in these phasic increases of activated microglia, what happens in the brain is that the neuroinflammation becomes dysregulated. Microglia eventually become exhausted, lose their capacity to efficiently phagocytotize the cellular debris. And then they start to morph into more of a inflammatory phenotype. And when this occurs, the outcome is dysregulated overexpression of inflammatory cytokines, primarily the soluble form of tumor necrosis factor, so soluble TNF. And soluble TNF is what we've generally referred to in the past as TNF alpha, right? So this overexpression of TNF alpha, soluble TNF, in the brain, primarily coming from activated microglia, leads to overactivation of inflammatory processes from other cells downstream, and you get what's called unresolving neuroinflammation. So the natural process that would repolarize this inflammatory signaling to beneficial becomes dysregulated. It gets to be out of control, and now we have um, proliferation of neuroinflammatory signaling, leading to neurodegeneration downstream.
Gotcha. So the TNF or inhibition of TNF kind of stops that whole process.
Yeah, so right. So back to activated microglia, because this is the primary focus of this paper. Activated microglia expressing soluble TNF above a constitutive level is the primary driver of the dysregulated neuroinflammation downstream. So to inhibit this process, you have to be very careful. What you want to do is take away the excess soluble TNF without inhibiting the TNF signaling within those pathways that is actually required for repair, right? So the other receptor backup, soluble TNF signals primarily through TNF receptor 1, right? So there's two primary receptors in the TNF signaling process, TNF receptor 1 and TNF receptor 2. TNF receptor 1 is associated with neurodegeneration, cell death, and frank neurodegeneration once it becomes dysregulated. TNFR2, on the other hand, is associated with cell survival and repair, right? And both of these processes are going on at the same time. The difference is that TNFR2 does not require activation by a soluble TNF expressed by any cell, and in this case, activated microglia. So you can take away the excess soluble TNF without inhibiting the beneficial function of TNFR2. And that's where this elegance comes in. Because of the mechanism of action of XPro 1595, you can give this drug peripherally through subcutaneous injection. It crosses into the blood-brain barrier at pharmacologically active levels and will selectively inhibit the inflammatory soluble TNF while maintaining the reparative processes that are dependent on TNFR2.
Looking at proteomic data, this appears to be consistent with other mechanisms in Alzheimer's. Can you discuss that a little bit?
Yeah, so this is this was not the focus of the paper by Botu and colleagues, but it turns out that they've actually illustrated a mechanism that is well recognized as one of the key players in this transition from initially beneficial glial activation to detrimental neuroinflammation. And that's a process that involves expression of soluble TNF from activated microglia in combination with interleukin-1, another cytokine, and a complement factor called C1qa, right? So these three combined are the mechanism that is recognized by the Alzheimer's community as the mechanism that is associated with activation of another glial cell called astroglia, and the activation of that cell, what we call reactive astrogliosis. That level of reactive astrogliosis is actually associated in correlation analyses from PET scans and biomarker studies. That level of reactive astrogliosis is what's associated with the initial cognitive impairment and progression of disease along the Alzheimer's continuum, right? So to put that in a nutshell, what these investigators have done is, in addition to showing that the TNF system is integrally involved in demyelination and remyelination, they've also illustrated, once again, a mechanism that has been illustrated by others showing that overexpression of soluble TNF, primarily from activated microglia, is the key signaling pathway that activates reactive astrogliosis, and the reactive astrogliosis in this paradigm is associated with cognitive decline and disease progression in Alzheimer's disease in the presence of the core Alzheimer's pathologies, both amyloid beta and hyperphosphorylated tau.
Interesting. So just bringing it back to remyelination, some of the other therapies are trying to stimulate myelin production. What is different in our approach?
Our approach is we're literally leveraging the natural process that exists, right? So as we age, our white matter becomes fragmented and has to be repaired. Unlike gray matter, white matter can be repaired and remodeled throughout the adult human lifespan, right? And that process is through remyelination, right? So the cellular machinery for repair of the myelin sheath exists throughout adult life in humans. And in the case of neurodegeneration, the inflammatory signaling from the neurodegenerative process, whatever that happens to be, whether it's head injuries or a disease like Alzheimer's, that neuroinflammatory signaling, the overexpression of these neuroinflammatory cytokines, impedes the ability of the myelinating cells to do their job, right? So the objective is to repolarize, in this case, the activated microglia, to their homeostatic activation phase so that they can continue to release trophic factors and other things that we won't get involved in or delve into today, but other cytokines and signaling molecules that potentiate the reparative process, right? So to say polarization, if you take away the excess signaling from soluble TNF, you're essentially polarizing the signaling towards that beneficial receptor, in this case TNFR2. And that's what these investigators have demonstrated.
Just bringing it all back to Alzheimer's here. Demyelination is a hallmark of Alzheimer's, right?
It's not necessarily recognized as one of the key hallmarks. It is emerging as a key factor, right? So the research that is ongoing is starting to show that this demyelinating process occurs very early in the progress of or the progression of Alzheimer's pathology. It's one of the earlier signals if you're looking for it and have the ability to measure it, which is difficult. But we do know primarily from postmortem data from autopsy studies that this demyelinating process occurs early in the continuum of Alzheimer's disease and worsens along the progression of the disease, and is driven primarily by the neuroinflammatory signaling, right? So whatever the stressor happens to be, right? Whether again it's a acute injury or an infection or the presence of a abnormal protein, all of these pathologies trigger neuroinflammatory signaling. Once that neuroinflammatory signaling becomes dysregulated and goes off the rails, it becomes compartmentalized within the CNS and doesn't resolve like it would, for example, if you get a bee sting on your finger. This unresolving neuroinflammation potentiates the overexpression of other neuroinflammatory processes downstream, and it's not specific to any particular disease. This is just a natural process, and that is the primary contributor to the neurodegeneration that marks the end stage of these conditions.
Parris, this has been highly informative. Thank you very much. I think we
Well, I'd
should.
love to be able to go into more detail, but it would take hours. So if you could, you may want to tell your listeners to stay tuned, and we'll be coming out with more material that will, flesh this out again in more detail and provide, better demonstration of what we're actually saying. But the key points, again, the key takeaway here, I want to emphasize this: the mechanism of action, of XPro is elegant, is very elegant. We're leveraging a natural process. We're not impeding a natural process, right? We're leveraging the natural reparative ability of the cells in the human brain to potentiate that maintenance and repair of neuronal tissue throughout adulthood.
Awesome. Well, appreciate you taking time, and look forward to more data and more opportunities to have you on in the future.
Thanks, Dan, and look forward to talking to you as well.